Hi everyone, I'm Anna Köck, and representing our collaborative work with Lausanne, I'm excited to share our paper on how the MYCT1-IFITM2 complex restrains endothelial energy waste. To put our work into context, think of the endothelium as more than just lining a blood vessel. It's a massive network over 1 trillion cells covering our entire vasculature. But these cells aren't passive. They are active gatekeepers of whole body health. Our goal was to find novel regulators of the endothelium. We focused on MYCT1, a transmembrane protein that is highly conserved across species and expressed in the endothelium of every organ. Despite being so evolutionarily well preserved, its function was unknown. We asked, what is MYCT1's function in the endothelium? To find out, we deleted MYCT1 specifically in the endothelium of mice. The results were striking. Even on a high-fat diet, these mice had reduced weight gain. MYCT1 knockout mice had less white adipose tissue and smaller adipocytes. So, why was the lack of one endothelial protein reducing weight gain? We looked at the usual suspects, but it wasn't because they ate less or moved more. The adipocyte development was normal, and also the blood vessel density in the fat was unchanged. So behavior, adipogenesis, and angiogenesis were all off the hook. So how does MYCT1 as an endothelial protein regulate weight gain and white adipose tissue expansion? We turned our attention to nutrient-sensing pathways and found that MYCT1 deficient cells exhibit a pronounced hyperactivation of mTORC1 in response to amino acids. Similarly, in the aorta of our mice, this hyperactivation only occurred under fed conditions, indicating that this effect is nutrient-dependent. Very importantly, also adipose endothelial cells hyperactivate mTORC1 signaling in MYCT1 knockout mice. This suggested that MYCT1 normally acts as a necessary brake on endothelial mTORC1 activation. The next question was, how? By analyzing interacting partners of MYCT1, we identified IFITM2 and 3 proteins as key binding partners, whose interaction we also found to occur in human tissue. IFITMs are known regulators of endolysosomal trafficking, and we found that loss of MYCT1 results in IFITM accumulation, leading to enlarged endosomes and increased lysosomal degradation. These changes in endolysosomal trafficking then result in this excessive mTORC1 activation upon the loss of MYCT1. We confirmed this hypothesis by simultaneous knockdown of MYCT1 and IFITM proteins, which rescues the mTORC1 hyperactivation of MYCT1 deficient endothelial cells. In addition, we show that endothelial-specific deletion of TSC1, which is an inhibitor of mTORC1 signaling, this forces cells to be hyperactive. And this actually phenocopies the restricted white adipose tissue expansion observed in our MYCT1 model. So together, all these experiments establish the mTORC1 hyperactivation as the mechanistic link between endothelial MYCT1 and the adipose tissue phenotype. In short, we've identified the MYCT1-IFITM complex as a previously unrecognized endothelial regulator of systemic energy balance. When this endothelial brake is gone, the vascular barrier induces a futile use of energy. This creates a metabolic bottleneck that limits fat expansion. All in all, we found a new way the vasculature shapes our fat. Thanks for listening, and I hope you'll enjoy our paper.